Okay, so the second part of this lecture is going to be talking about the GSEA algorithm, or the gene set enrichment analysis algorithm, which many of you might have heard of before. So this was originally published in 2005 in this paper that I cited at the bottom if you're interested. And basically this algorithm looks at if a ranked list of genes is significantly associated with some biological function represented by a gene set. So do note that GSEA algorithm actually does take in a list of ranked genes, not just a set of genes. The original method, I believe, is available online from their website. I think you can also download the desktop software application. I believe there's also an R implementation of this algorithm developed by this team. Basically, a quick overview of this algorithm. So you have a ranked gene list, and you place this along an x-axis of a graph, and then you compare the ranked list to some annotated gene set. And then what you do is you compute a running sum, or a Brownian Bridge random walk. So you can kind of see on the right, you see that red line, that is the Brownian Bridge plot. And then you compute the enrichment score as the peak of that Brownian Bridge plot. And then finally, you can estimate the significance of the relationship between your ranked input list and the annotated gene set. And if this all sounds a bit complicated, I'll kind of go through the steps a little bit more in detail, but also I highly recommend you read the original paper if you're very interested in the specifics of this. So as I said before, you're going to have some type of ranked gene list. So for instance, let's say you're comparing gene expression in two types of samples. You have samples from class A and then samples from class B. And then you can generate on the bottom there a type of signature or some type of ranking where the genes are ranked by their differential expression values. And then let's say that you want to compare this to some gene set annotated with a known biological function. So for the purposes of this example, we're just going to say that the gene set we're looking at, or the pathway we're interested in, has these associated genes B and C. So once you've ranked the genes, you want to generate this random walk plot to compute the enrichment score. And this is an example of one from this paper side at the bottom, which is also a great resource for reading further about gene set enrichment analysis. So to understand what the point or the logic behind this random walk plot is, it's basically built upon this idea of Brownian motion, which I think at its very physical definition is the random motion of particles at thermal equilibrium. An example of this that is basically underlying the plot that we're going to build is, imagine you're starting at 0 on a one-dimensional number line, and you're randomly taking steps to the left or to the right. So each step you can either go left or right randomly. And so you can create this graph of the displacement from the original position of 0 over time. So on the left, you can see if you take a few steps, that might be what your path looks like. And then on the right, you'll see that the more steps you take, the more fluctuations you're actually going to get in the displacement. But you see that generally you are getting further and further away from 0 position. So the Brownian Bridge plot basically follows that same concept of random steps, except the start and the end points of this walk are actually fixed at 0. So you start at 0, you end at 0, but the steps in the middle are basically random. And once again, you can see that as we increase the number of steps we're taking, you see a lot more fluctuation. And then what is known as supremum is basically this maximum displacement of the walk over the Brownian Bridge. So here on the right, you can see that the supremum looks like it's positive. It's not always necessarily positive. It could be a negative value as well. And I'll talk a little bit more about what that means for enrichment analysis results. But basically, getting back to how that all applies to the gene set enrichment analysis algorithm, if you're really interested in the mathematical specifics, again, highly recommend you read the original GSEA paper, or you can read the paper that I've cited at the bottom of the slide from which these examples come from. But generally, the distribution of the displacement in a random walk, in a true random walk, will follow a normal distribution. And then because it follows a normal distribution, you can actually look at the cumulative distribution function of this normal distribution, which is the expected CDF of a random walk. And then you can compare that with the CDF that you observe from the walk you generate from your comparison data. And this, in the GSEA algorithm, how they evaluate this difference is basically a version of the Kolmogorov-Smirnov test, which you can also Google if you're interested in the mathematical specifics. But essentially, the gist of it is that if you look at the two plots on the right, you can see that you have the expected CDF of the data in red. So that is generally the CDF of the normal distribution. And then you have what would be the CDF of your input data in blue. And you can see that when the data is consistent, when your data is consistent with a true random walk, your CDF will be consistent with the CDF of a normal distribution. And you can see that you have a pretty random walk at the bottom. So you're staying around 0 here in your random walk on the bottom. However, you can see that when your data is actually not a true random walk, as in there's some type of bias, you'll see that the CDFs will actually differ, and that'll also be reflected in the random walk plot that you generate. So you can see here on the bottom right, this random walk that you've generated actually seems to bias towards the positive side of 0. So that is all the very specific mathematical details. Again, if you're very interested, read the original papers because they explain it probably a lot better. But returning to our very basic example, so we can start generating the random walk for this data that we have. So basically, we're going to start walking down this list of ranked genes with the first gene at the first position. And so if we find that if the gene that we're looking at each step is not in our comparison set, then you can increment or actually decrement by this value, which is just the square root of g over n minus g, where g is just the number of genes in your comparison set, and then n is the number of genes in your data. And so you can see because these first 3 genes, as we're walking down the list, D, A, and E are not in the comparison set, we're actually going to decrement steadily each time. However, if the gene is in the comparison set at a given step, then you actually want to increment by that same value. And here you can see that because C and B are both in our comparison gene set, we're actually incrementing the graph at those steps. And then so obviously this is a short example. You're usually not going to have just a gene set with 2 genes and data with 5 genes. But in this example, you can see we've generated this full random walk or running sum graph. And you can see that the supremum of the random walk, which is the enrichment score, is actually this negative value that's at this most extreme point of the graph. And in general, if you get a positive supremum or enrichment score, this indicates that the genes in the comparison set tend to be upregulated in your data. When the ranked list of genes that you're looking at, so the input list that you've generated, if it's from a GWAS or ChIP-seq study, note that you really only care about the positive supremum because you're only looking for genes that are associated with either the trait or the transcription factor that you were looking at. A negative supremum, like the one that we got in our example just now, this indicates that the genes in the comparison set tend to be downregulated in the data, or they were downregulated in class B compared to class A. And then, of course, if you have no significant deviation from y equals 0, or your line is pretty horizontal, this generally indicates that the genes in the comparison set aren't really significantly differentially expressed in either direction in the data that you input. So for some more realistic examples from real data, I've included these figures from the original GSEA paper. So here you can see a plot, a random walk plot when you're looking at genes that are ranked for differential expression between male and female lymphoblastoid cell lines. And then the comparison gene set is chromosome X inactivation genes. As expected, because you're comparing female and male, and those chromosome X inactivation genes are associated with sex, you can see that there's a positive suprema, which means these genes are typically significantly enriched in the female samples, which is the expected result. Now let's see a different example. So now we're comparing the same genes that are differentially expressed between male and female cell lines, but now you're comparing against this vitamin C import pathway genes. So these genes are not really sex-based, and you'll see that based on the random walk plot, the genes that are in the gene set are spread pretty evenly across the ranks, so there doesn't appear to be any significant enrichment. So I mentioned that if your plot is pretty close to 0, you don't have really any significant enrichment, but you can actually specifically estimate the significance using the GSEA algorithm instead of just kind of eyeballing it. And how the algorithm generally does this is it will randomly swap the class labels for each of the sample and repeat the process that we just walked through, basically computed the enrichment score many, many times. And then ultimately, you can compute the significance by calculating how many times the enrichment score that you observed from the actual data you had is larger than the random enrichment scores that you computed. So in general, the GSEA algorithm is a very well-known and useful method for computing enrichment analysis for a ranked gene list derived from some comparison between two conditions. The algorithm produces this enrichment score based on the supremum of a Brownian Bridge random walk plot. The significance of the results can then be quantified by just comparing your observed enrichment score against repeated random permutations of class labels and the scores that are produced from those permutations. And then finally, in terms of implementations, I do want to mention that since this class mostly works in Python, as far as I know, there are 2 Python implementations of GSEA algorithm right now. There's GSEA Pi, and then there's also the Blitz GSEA package, which was actually developed by Dr. Alexander Lockman. And at the end of this lecture, I will kind of run through that notebook if some of you have already taken a look at it. But that notebook does give an example of how to use both of these packages for data.